Well, the traditional treatments for cancer, and including breast cancer, have been what I call the sledgehammer approach. So the treatments are based on substances like Taxol that interfere with cell division. So when a cell divides, its spindle has to break down and reform so that the products of mitosis can form the two separate cells. But some of the cancer drugs target this process, and they prevent the transition from polymerization to depolymerization of these microtubules that comprise of the spindle. So what that means then is the cancer treatments are going to target all dividing cells in the body, not just the cancer cells, because Taxol is non-specific. So as a consequence of that, people who are on these treatments get really unpleasant side effects, like their hair falls out, they have gastrointestinal problems because the drugs are targeting the rapidly dividing cells that are normally in the body, the skin cells, the gut cells, the hair cells, as well as the cancer cells. And so those treatments are often for a sort of competition between the side effects versus the benefits. And there are so many side effects, obviously, of interfering with normal cell division that you have to be careful that that's not going to affect the patient. So a growth factor is a small molecule that stimulates the growth of cells, either specifically, in other words, it targets one cell and not another, or generally. So if you have an injury to your skin, then you know that over time you get a wound that heals. And so the processes that involved in the wound healing involve growth factors, like growth factors that affect the epithelium or the outer layer or the skin, and it causes the cells to proliferate and to replace the dead and dying cells. So growth factors are really important for all cells in order for them to actually progress through the cell cycle. They make cells move from one stage of the cell cycle to another. And if you're going to have cells reproducing because they've been worn away or they've died or they've been injured, then you need growth factors for this. Well, the new treatments, I suppose, are based on an understanding of the molecular biology, because if you don't understand what's going on in the cell, you can't really design specific treatments. So I think what we're finding today is that treatments for cancer are based on the type of cancer. So we try and target the drug to the cancer cell and not to the other cell. So that really requires that you know what the properties are of the cancer cell and what markers it has. So then you can build up a drug delivery system that only goes to that cancer cell and not to other cells. But in order to be able to do that, you've got to understand about the molecular biology of the cancer cell in the first instance. The treatments nowadays are actually targeting the cells one of the ways is just using the same drugs but targeting them to the cancer and not to other cells. So that's sort of an antibody approach to cancer treatment. So that's one way. But then there are other approaches where we can try and deliver internal components that will interfere with the pathways that the cancer cell has, like small nucleic acids that will get in and interfere with the processes of the cell that are causing it to be a cancer cell. So it's quite complicated, but with the genetic knowledge, we can use that to try and target the pathways going from the genes to the proteins that are, and try and switch them off in the cells that have got the cancer process going on. Well, I suppose whenever we look at a disease or a process in a cell, it's like a jigsaw, and we can never really find all the pieces, but we can try and put together the big picture. So the way I see science or any area in science is like a jigsaw puzzle where you fit together the pieces and you get an overall picture. And this applies to cell biology, where we have a certain understanding of how cells work. I mean, most students would know about the DNA, the RNA, and the protein, but then the regulatory processes by which you go from the gene to the RNA to the protein are extremely complicated. And we're trying to understand those so that then we can maybe look into managing maybe the genetic control of a cancer cell. So any information about new molecules that are involved in these processes will be useful. It will fit into the jigsaw puzzle so that we can understand what's going on. But it's a bottomless pit in a way, because no matter what you find, then there seems to be another layer of complexity on top of that. And if you weren't a scientist, you probably get a bit frustrated with that. But I think that's the reality of life as we see it. As scientists see it, that it's complexity and we have a limited capacity to understand it, but we can certainly make headway. And you have to say that we've done quite a good job in treating diseases and building bridges and getting to the moon and whatever, even though we only have a very limited understanding of the whole picture. Yes, well, I'm working on another cancer at the moment called a neuroendocrine tumour, and with an oncologist, we managed to get a National Health and Medical Research grant last year, and this is enabling my oncologist colleague to treat patients with a new treatment where he's put a drug, a drug for treating cancer, knocking out cancer cells, to an antibody, and that will target the cancer cells. So he's doing that with the patients, and then because not much is known about this cancer, they don't know how to diagnose it, they don't know how to treat it properly, and the other thing is the patients develop drug resistance. So our laboratory-based study, which is called a translational study because it's meant to translate back to clinical usefulness, our translational study is looking at novel markers, which we found, and then trying to identify what the behaviour is of the circulating tumour cells, so that cancer cells get in the blood, travel around, and we get patient blood and we can identify those circulating tumour cells. They're quite hard to identify. I mean, my research fellow on this is doing a great job. And then we can correlate the profile of markers in the tissue and in the tumour cells with the clinical picture. And hopefully that's going to provide a lot of insights for diagnosis, but also on how effective treatment is. And on the other hand, we want to know why some cells develop drug resistance. Why is it that some cancers become resistant to drugs, and that means the patient's life is cut short, whereas other types of cancers, they can be treated for years with drugs and without any problem, without the cancer taking a grip. So I think that's a very important question to answer. Well, I have to say that scientific concepts can be hard, but it's just the reality, and you don't understand things the first time, you just have to persist. And I think it helps to read a lot of different ways of, different approaches to explaining it. And if you're not sure about one textbook explanation, then go to another textbook, or even better, to ask a person that has a really good understanding of it. The fact is that biological pathways are incredibly complex, and you might have seen the metabolic pathway with like 1,001 arrows going there and everywhere, and agents and products and reactions everywhere, but that's the reality of it, and we just have to come to terms to it. But it doesn't happen overnight. It takes quite a long time to understand, to get a basic grip on it. It took me a long time, and what I found is that I had these patches of knowledge all over the place, and it wasn't until many years that I managed to link them all together. So now I've got an overall picture of how I see a normal cell, how I see cells in certain types of diseases. But It doesn't come overnight. It's quite a complex thing, but it's fascinating. It's absolutely fascinating. And if you have a good textbook, it makes it extremely interesting and worthwhile to read.